Today it's my great pleasure to talk with Professor Alex Verkhratsky. He's a professor of neurophysiology at the University of Manchester in England. He's also, I think, a professor of the Basque Foundation for Science, which is kind of interesting. And he's an expert on glia, which are all the cells other than nerve cells in the nervous system, brain, spinal cord, and so on. And he's pretty much devoted his whole career to understanding these cells that have been grossly understudied. You know, neurons are kind of attractive because they're forming synapses and they're form this circuitry, but these other cells that he's going to talk about with me today do very important things. The neurons would not be able to do what they do without these cells. So Alex, welcome.
Thank you very much, Mark, and I'm very pleased to be with you, and thank you very much for this invitation. And of course, I mean, with you, for me, one of my things which I'm really very much interested throughout of my life is aging. And of course, I was following your seminal works in aging for many years, and that gave me lots and lots of exciting moments and everything. So yes, GLIA. It's very interesting because whenever you open almost any paper in the world, it will say GLIA was understudied. In reality, it's completely different because when the GLIA began in the 19th century, it was overstudied. And why it was? Because most of neuroanatomists were medics and they were pathologists. And whenever you open the brain, which has a pathology, you see that there are not neurons which are changing. Neurons are dying. It's glial cells which are changing. Very early, already in 1860s, 70s, people realized that those cells which are glia, which were called neuroglia in those days and so on and so forth, these are pathologically relevant cells. And up to probably 1920s, 30s, these cells were very much in the focus of attention. And I think what signaled the change was electrophysiology. Because of course you take an electrode, you put it in neuron, and you are fascinated with your oscilloscope, which does all the things for forever. You put it in glial cell, and nothing.
So, well, but you found there is something.
Yes.
So you were born in Ukraine, is that right?
Yeah, I was born in Ukraine, which at those days was still Soviet Union. And I was, in a sense, very lucky because after having medical degree, I studied electrophysiology in what was called Bogomolets Institute of Physiology in Kiev. And that at those days was really at the forefront of quest for ion channels for patch clamp. And the Herbinger of patch clamp was made there. So they made some plastic system for intracellular perfusion. And for years when I was there, and I began in 1979 as a student still, as an undergraduate student, the institute was full of foreign people. And I knew personally Bert Sackman and Erwin Mayer and Alain Marty and Hamel, and so everybody, they were there. And that, of course, very much influenced my future career.
So Mayer and Sackman got the Nobel Prize for developing patch clamp, but they were not in Germany? Yeah, they were in Germany.
Oh, yes, they were in Germany, but they were at those days actually in Göttingen. And then Erwin stayed in Göttingen till the end of his scientific career, and Bert went to Heidelberg, where he got Max Planck. But during this late 70s, early 80s, all of them were visiting Kiev laboratory. And that's how I knew them. And really, I was incredibly lucky in my life because I met very many top scientists when I still was a student, which obviously very much shaped me, I would say. It was very important. One of my absolutely closest friend was, for example, Jeffrey Bernstock, who discovered cholinergic system.
Wow.
And that was an amazing person, amazing personality with lots of knowledge, and as a person, extremely warm and extremely nice. And then, of course, I mean, I cannot not to say a couple of words about this war, which is now conflagrating the Ukraine and everything. And in my view, this is really a sort of turning point in our history because it's a new Hitler. It's a new fascist country which tries to put itself on the top of all our life, in a sense, and all our beliefs and everything. And the outcome of this war could probably shape the rest of the century. So, of course, I'm trying to do all I can in order to help Ukrainians and in whatever possible ways. But clearly, it's a very bad time for science. I mean, more or less, science doesn't exist, which is understandable because during the war, all the resources go for the front line. So it's absolutely terrible.
Yeah, they're gonna maybe lose, I don't know, possibly a generation of new, the students who would have been going into science, PhD or postdoc, don't have that opportunity right now.
Well, I mean, in principle, of course, the scientific history of Ukraine was rather sad because the country didn't really support science for the last 30 years. And let's say my generation, from the people with whom I was studying in 70s, 80s, 90% cerebral.
Yeah.
So most of people left. And that is understandable because if you're a scientist, you need some conditions to do science.
Yeah.
And in Ukraine and post-Soviet times, these conditions simply didn't exist. So in a sense, it's a multiple trauma because not only the scientists left, but all educational system obviously deteriorate. And I could not imagine how to get it back. And so presumably it would be international effort, I don't know, but it will be very, very, very difficult.
That's unfortunate. I was watching the world track and field on television. I was a runner and followed that, and Ukraine had a high jumper that got a gold medal, a woman from Ukraine. And that was very kind of emotional.
Yeah, it is. Yeah. I mean, the whole war, of course, is incredibly emotional. And since the beginning, every morning, what I do, I of course open the local sites and see what is going on. And all these bombardments and all these missiles which are landing in the cities and killing civilians, it's just part of Russian mindset. As I said, I think it's a criminal country which doesn't deserve to remain in the history.
Yeah. Okay.
So Russia delenda est. Okay.
So let's get to the science. You initially, so you learned the electrophysiology from the people who developed these methods, and then initially you did some experiments in neurons, but then quickly.
No, in cardiomyocytes.
Oh, in heart cells.
Yes, heart cells. My PhD was cardiomyocytes, and that was this very exciting time when for the first time you can get isolated cardiomyocytes which are functional. And that was done for the first time by Gary Tisenberg in Germany.
So are these beating? Are they moving?
Oh, yes. They were real, and true so cardiomyocytes.
You're putting an electrode into a moving
and you can initiate contraction, So, and Gary Tisenberg created a special media, if you will, for these cells to preserve them, and he called it Kraftbrühe, which means the brew of power. So that was a series of 3 papers in Flüger's archive, and we followed that, and I did all these cells. So my PhD was actually on cardiomyocytes.
Okay.
On sodium cards. And then, of course, this is, as you know, the serendipity of life. In Kiev, where I was in those days, it had quite a good relations with Germany. and we had meetings with German scientists. And one of those meetings in mid-80s, I met Helmut Kettenmann, who was in Heidelberg at those days, and he was maybe one of 5, 6 people in the world who were studying glial cells.
Yeah.
And he invited me, and I came to him, and that was the end of it. So I was completely captivated, and since then I could not stop looking for these cells because it's very, very interesting.
Yeah, and at that time it wasn't thought. So you mentioned sodium current. So sodium's high concentration outside the cell, low inside. So, and there's channels in the membrane that they're closed, and then when they open, sodium comes in, so you get a voltage change across the membrane. And at the time you started working on the glial cell, that was thought they're not excitable, and maybe people said, why even bother trying
why
to
bother?
Yeah.
Absolutely. That was, we had our first meeting of glial people of Europe. There were maybe 30 people. And that was the end of it. So in a sense, again, it's my luck because I know everybody personally who was studying glia at those days, so from the very beginning. And again, from the very beginning, it was quite unusual because we really all found that glial cells do have channels. Another issue, they have them in different numbers and different densities, which do not allow them to produce what is called action potential.
Yeah.
So they cannot do regenerative response, but they do have lots and lots of other responses. So of course they're electrically non-excitable, but they're very excitable in other terms.
Yeah.
And the substrate of their excitability is actually intracellular. So for neurons, the substrate is plasma lemma, is plasma membrane, right, with all the channels. For glia, it's inside, it's innards of the cell, if you will, in which different ions are coming. And of course, calcium would be number one, which was very much studied. But second ion, which is incredibly important, is sodium. And sodium does come to astrocytes in big numbers. And this sodium actually coordinates astrocytes with neurons. Because at the very core of astrocytic function lie the so-called transporters. So these transporters are sitting in plasma membrane, and they transport molecules. Yeah. And for example, the most important molecule of our life in the brain is glutamate. Yeah. So neurons release glutamate, but they don't know what to do with it. So nearby, you have an astrocyte, which sucks this glutamate with the help of transporter. And those transporters are many, and most of them are sodium dependent.
Alex, so you mentioned glutamate. Since I retired from that, I wrote a book. Yes. This came out.
Mark, actually, I just received this book 2 days ago, and I'm looking forward reading it.
Oh, really? Okay.
I mean, I ordered it and took a little while, but and yes, I mean, glutamate is one of the.
But anyway, so the astrocytes, they can have increases in calcium in the cell, increased sodium, and then in the membrane there's this sodium calcium exchanger, and then neurons respond to astrocytes respond to glutamate.
Yeah, so they respond. You see, it's very, very interesting, and again, it just shows how intricate is the neural tissue. Because we cannot say these are neurons, these are astrocytes, this is microglia. They are all coming together, they work together. Now, neurons, for example, they cannot produce glutamate. They don't have an enzyme.
Yeah, that's true.
Astrocytes do produce glutamate, and this glutamate is what neurons do and use for their synaptic transmission. But it's, again, now a big question, how to transport this glutamate to neuron. You have to give it out. And glutamate, as you certainly know, is a toxin. It's a fantastic poison, which kills everything. Yeah. So what nature did, nature made a new enzyme, which is called glutamine synthetase. And this glutamine synthetase takes a glutamate and converts it to glutamine, which is neutral, which is perfectly all right. And then this glutamine goes to neuron, and neuron now makes glutamate and GABA. So if you stop astrocyte, there is no neurotransmission because neurons cannot produce glutamate. So that, I think, is a wonderful example showing that the nervous tissue is really cooperation. And we cannot say this class of cells works independently from that and from that. So it all comes together. It all what is called active milieu of the nervous tissue.
Can you talk about this coming together? So you're talking about physically, I mean, being membranes being right next to each other, maybe a good, why don't you talk about the neuronal synapses and how these glial cells are intimately associated with those synapses, and how this is important in regulating the function of the synapse.
So again, this is incredibly interesting because, of course, when we start to look into most of pictures on the internet, which depict the brain, that will be only neurons, and in between you will have axons, and something is running around and everything. That is, of course, not the case. So what we have a incredibly densely packed tissue, of which about maybe 10–20% is extracellular space. Everything else is stacks of membrane of a very, very tiny thing. So, for example, one synapse would occupy probably, I don't know, a couple of less than a couple of cubic microns. And around every, or most of the synapses, at least in cortex, they're astrocytic membranes. And they make like a protrusions, which we call leaflets. And those are most amazing structures because their thickness is about 100 nanometers. So imagine a single membrane is about 20, So you have two membranes, which is already 40 nanometers, and 60 nanometers in between. So it gives an incredible surface to volume ratio. So it's a huge membrane with a very tiny volume. And this membrane is absolutely packed with transporters. So the density of glutamate transporter is about 10,000 molecules per square micron. This is a density of sodium channels in the node of Ranvier. So that gives you an idea. So in our brain, 1.3 gram of the whole weight, which is what? 1.5 kilograms, is glutamate transporter in astrocyte. It's incredible. It's an amazing molecule which is everywhere and which is life-saving, because if you don't control glutamate, it's all dead. And in that sense, if we start to look what is called neurodegenerative diseases, most of them are at the end converging into astrocyte, which stops controlling glutamate. And then neurons die. So in a sense,
one point about that too, Alex, is that there's some naturally occurring excitotoxins. So these are molecules that activate glutamate receptors, canic acid, domoic acid, and so on. And in contrast to glutamate, those excitotoxins cannot be taken up by this glutamate transporter.
They can, but much, much worse.
So, but they stay outside the cell much more.
Well, I mean, it was an example, I think, in one fisherman village in Canada, somewhere near Halifax, when they were eating mussels. And these mussels were feeding on some seaweed, which produced domoate, which is analog of kainate. So these people ate these mussels, and half of them died, and another half became neurodegenerate immediately. That is exactly what happens, because there is no natural system to protect. So if you again look into the synapse, you have this incredible membrane which sits here, and this membrane does everything for synapse to work. So in that sense, astrocytes are par excellence a homeostatic housekeepers of the brain. And without them, there is no brain.
Yeah.
And I think this is really very important to understand, that all these things are working together, and neuron without astrocyte cannot do anything. Astrocyte without neuron can actually survive probably, but again, it wouldn't produce salts. So,
yeah. And also what, why don't you talk also about the energy metabolism, right? That these glial cells play an important role in the energy current utilization in the brain.
Yeah. Well, it's a very, very interesting topic, which for a while, everything was seemingly very clear and very simple. So the idea was glucose comes to the brain, and of course, glucose is a main energy substrate from which brain makes ATP. And half of it goes to neurons, half of it goes to astrocytes, but astrocytes don't use it. They convert it to lactate, which goes to neurons. So it was believed that about 90% of energy is spent by neurons and only 10 by astrocytes. And these astrocytes were consequently considered to be glycolytic cells, so no oxidative phosphorylation of very little, doesn't matter. It turns out that when we have neuronal activity, the neuronal activity is, as we already discussed, is there are channels. So there is sodium channel, sodium goes in, but then you need to come back, and that is potassium ion, which goes out.
Yeah.
And this potassium goes out, and it has to be removed, because otherwise it will change all biophysics and neurons will stop working. And neurons cannot do it because they don't have a proper enzyme. And the proper enzyme is sodium-potassium pump.
Yeah.
And there are three isoforms of sodium-potassium pump. Two are in neurons. They're called alpha-1 and alpha-3. And one is in astrocyte. It's alpha-2. And the difference is that neuronal ones are having Kd for potassium like 0.5 millimole. So in physiological potassium, they're saturated. Astroglial has Kd about 3.6 millimolar. So it's completely non-saturated. And whenever potassium goes up, it immediately activates the pump. But pump needs energy.
Yeah.
And it turned out that at least 30, if not 40% of all energy of the brain is actually consumed by astrocyte to take away this potassium. So now we have what is called great metabolic revolution. So we realize that astrocytes are actually major energy consumers, and they need, of course, oxidative phosphorylation to do that. They do have glycolysis, and what is good for astrocytes, if needed, they can switch. So if there is ischemic period, neurons, of course, die very rapidly. Astrocytes can survive. And when they go into this glycolytic pathway, the intermediate of glycolysis is lactate. And lactate is actually a very good energy substrate because it's ready to go to TCA, to a 3-carbon cycle, acid cycle. So astrocytes can supply these poor neurons with a very good energy supply, energy substrate. The whole story...
Alex, what about ketone utilization by astrocytes? There's a lot of interest in now in neurons being able to use ketones and...
Yeah, that's a little bit away from my main line, but astrocytes actually can fix ketones. So they can take them up and they can use it as energy substrate without making any harm.
Yeah.
And when it comes to all this high-fat diet, of course, we all know how bad it is and don't eat burgers and blah, blah, blah. But if you look into an early development, a young brain, probably 50% of energy takes from fat.
Yeah.
So actually for developing brain, fat is absolutely needed. It's when we become old, the fat becomes harmful. Yeah. So it just shows how nice is biology, that it's never, it's not a black and white. It's never a one particular thing. It's a big continuum of many, many responses and reactions which are making us what we are.
Now these, the glucose, the ketones, they come from the blood essentially. Can you talk a little bit about the role of these glial cells in and coupling activity to the blood flow?
Yes, that's an. I have a feeling, Mark, that probably we need 10 podcasts to know
Yeah,
those
I
things.
know, I know.
So if you think about the brain, of course, the brain is very different from anything else because it has what is called blood-brain barrier. So what does it mean? It means that blood vessels are not particularly permeable. So they have some fence which limits the permeability and which chooses what to go through. And that is a very interesting story because, again, if you go anywhere to any book, whatever, you will find out that that was a Paul Ehrlich who discovered blood-brain barrier. That's wrong. He actually was completely against the idea. He did the experiment. An experiment was, every beautiful experiment is simple. So what Ehrlich did, he took some blue dye and injected it into the animal. And everything was blue and brain was white. And that was already from that, you can say that that was a barrier. Now, Ehrlich thought he was physicochemist. He thought, no, it's a different affinity of tissue. But one of his pupils, Lewandowski, Max Lewandowski, and also Erich Goldman, they repeated the experiment from another side. They put it in the brain. Now, brain is blue and the rest is white. And they start to come with an idea of the barrier. But the actual term was introduced by Lina Stern, who was working in Geneva in 21. She made the term hematoencephalitic barrier, barrier hematoencephalitic. So that's how it's done. So this barrier, again, if you look to the textbook, it's all very simple. So what we have endothelial cells. And every vessel in our body is covered with endothelial cells, the wall. But only in the brain, these endothelial cells are connected with a very specific proteins which are called tight junctions. And they make a barrier. So really, water cannot penetrate through this barrier. And then these endothelial cells have many, many transporters which say, oh, I need a glucose, I take a glucose and put it in. Or whatever drug comes, and the endothelial cell doesn't know that this is a drug. And it says, no, no, you don't go in. So many drugs which we use cannot. Again, the whole story is much more complicated. So we have these endothelial cells. And then we have a wall of a vessel, which is endothelial barrier. Yeah? Then, of course, there are some smooth muscle cells or pericytes and capillaries. And then we have a space. And it's called perivascular space. It's a space between the vessel, between the blood vessel, and parenchymal part of a blood-brain barrier, which is a specific process of astrocyte, which makes an end foot. So we have blood vessel, perivascular space, and end foot. And this perivascular space is full of cerebrospinal fluid, which enters the brain in paravascular route. And then it all comes to a capillary. And in capillary, this all disappears. So instead of two basement membrane, we have one. Here are endothelial cells, here are astrocytic end foot. So it stops the channel. And that somehow, we still don't know how, creates a driving force for all this liquid to go to the brain parenchyma, to circumvene the capillaries, and to come to another perivascular space in veins. And that is what is called glymphatic system, because brain doesn't have lymphatic vessels. It has instead these perivasculars. And that is of paramount importance to clean up the brain. Now, how does it work? We still don't know, but what we know, that if we knock down a specific channel, which is called aquaporin, it's a water channel, which is localized only in this perivascular, end feet of astrocytes, it stops working.
You know, these aquaporins, the aquaporins are interesting. Peter Agre, who's at Hopkins here, he discovered them and got Nobel Prize. And because it, you know, you have this lipid, this fat membrane, right, that water can't go through, but yet somehow water must be exchanging. And But I was getting more at, so that you talked about the blood-brain barrier, but I was wanting to talk more about the coupling of neuronal activity to blood flow. And
yes, it's another function, which again, it's still quite controversial because you see, most of experiments which we do in ex vivo preparation. For example, we make a slice of the brain.
Yeah.
If you make a slice and put it under microscope, you can see the capillary. And you can do certain things, and this capillary can change the diameter. But what is absent is a continuous pressure, because you don't have a heart, you don't have a system.
Yeah.
And this pressure probably is very, very important for the whole things. But nonetheless, the idea is quite interesting. It comes again from experiments in rodents. And if you go to the cortex of rodents, of mouse or of rat, you will see that these astrocytes are sitting sort of individually, they don't overlap. So they sort of parcellate the whole tissue into individual bits and pieces. And within this individual piece, you have an astrocyte, and this astrocyte sends at least one, or usually 3, 4, processes to the vessel. This sort of end feet, And the idea is that this astrocyte with end feet and with everything which sits within this territorial domain produces an independent unit, which is called neuroglia vascular unit. And within that, of course, this astrocyte can sense neuronal activity, can secrete something, and somehow change the diameter of a capillary. And that all is part of what is called functional hyperemia. And functional hyperemia is increase in blood flow in the part of the brain which is active. And amazingly enough, it was discovered back then in 1880 by Italian guy Massi, and then it was repeated by Sherrington in Cambridge. So it's 130 years old, the whole idea. All is good and nice, but so far there is one paper which was published about 2 years ago and which still sits in bioarchive, the preprint service, which gives me an idea that probably these people try to push it to a very high quality journal, and it doesn't. So what this paper did, it's very simple in a sense. It's a full, complete electron microscopy of a cube of human cortex, 1 by 1 by 1 millimeter. And they describe every cell there. So they have neurons, they have microglia, they have astrocytes. And it turns out that in this particular piece of human brain, astrocytes are completely, totally overlapping. They don't have individual domains. And if this is true, and of course, as I said, it's one preprint, nobody repeated it, it's still a preprint, but its morphology, it's very difficult to falsify morphology, right?
Yeah.
If it's true, then all our knowledge from rats and mice is irrelevant.
And this, so you describe these astrocytes, having these branches and then these end feet, and then some of them associate with synapses, some of them associate with blood vessel. But that when we did a lot of cell culture work early on, actually all the way through, and we'd take whatever, newborn mouse brain or rat brain and homogenize, separate the cells. If you put them on a plastic surface, you get these very flat cells that really don't have many processes, and those have the markers for these astrocytes. But then look in vivo at these astrocytes, and they have amazing branching morphology, as complex, or in some cases, in many ways, more complex than the neurons. And then the other thing is these astrocytes communicate with each other. You talked about calcium. Can you talk a little bit about this notion that astrocytes may play some role in long-range communication you know, because they're communicating with each other, maybe throughout the whole brain.
That's one of the ideas, which is quite old and which is called panglial syncytium.
Yeah.
For that, of course, we have a clear-cut structural basis, because what astrocytes have, which neurons mostly don't, some of neurons, of course, do, astrocytes have specific molecules which are called connexons. So what are connexons? They are quite big proteins, and each connexon is sitting in a membrane of one cell, another cell. And if they come very close, the gap about 2 nanometers, those connexons are aligned and they are forming one single intercellular channel.
Yeah.
And it's a very big pore, so it can pass molecules with a molecular weight to, I don't know, 1 kilodalton. So IP3 can go through, cyclic AMP can go through, metabolites can go, even vitamins can go through, so many, many things can go through. And of course, ions can go through. And that is the basis for this idea of glial syncytium.
Yeah.
And amazingly enough, when the whole story began about the brain back then in 1870s, 1880s, there were two major theories. One theory was reticular theory, which regarded the whole brain as one entity, that everything is internally connected. And the second theory, which is usually linked to the name of Ramon Cajal, looked at neurons as independent entities, which do not connect physically, but which do these synapses. As always, everybody was correct. So we have half of the brain reticular and half of the brain neuronal. And what is very interesting, if you put astrocytes in culture, as you started, and I think culture work was absolutely instrumental at whatever point. Although, as you mentioned, when you put astrocyte in culture, it's not astrocyte anymore. It's something different. It's a relative, probably a cousin, but it's a very different. If you put these astrocytes in culture and they grow, and eventually culture becomes what is called confluent. So they are all facing each other, so they have borders. And then all these astrocytes make these gap junctions, connexons, and they are connected. And if you take an electrode and poke a single astrocyte in the very middle of it, it will trigger calcium signal in this astrocyte. And then you will see beautifully the wave of calcium going around. And it can go through the whole of the calcium.
Yeah.
And again, there are different mechanisms. It could be gap junctions. It could be also some releasable messengers. It doesn't matter. But it definitely goes for millimeters. Yeah. And that was an idea about a long-range signal. Now we come in vivo brain. And we still don't have really clear-cut idea whether these calcium waves exist in the in vivo brain.
Yeah.
Because usually if you open the skull and start to look into astrocytes, say in motor cortex or in sensory cortex, yeah? And something happens, locomotion or visceral stimulation, or external stimulation. Usually these astrocytes do respond in synchrony. So they, all of them, which you can see in the field of view, go up with calcium and go down with calcium. And what is absolutely amazing, if you look into locomotion, for example, and of course, locomotion is also sensory information, so it's impossible to talk about just simple locomotion. The neurons would do calcium signaling all the time. They will fluctuate because, they are excited and open channels, calcium goes in. The astrocyte will produce one single big response to the onset of locomotion, and that will be it. So this big response does something which probably coordinates astrocytic support with neuronal activity. So again, fortunately, now we realize that we have to go in vivo. Because even if you do brain slice, it's an incredible trauma. And of course, when you slice the brain, all these interleukins or cytokines or you name it, they go over the roof. And there is lots of damage. So we have to go in vivo and we have to go to awake animal because anesthesia, it's even worse. It's more or less toxical coma. And but interpreting the data is, of course, very difficult.
You know, Alex has, I haven't been following this very well. Have people been using optogenetics to put channels in astrocytes?
Oh,
Yeah.
yeah.
And 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 then do calcium imaging?
Oh, yes.
Yeah.
They can do calcium imaging, or they can induce calcium signal in astrocytes and then look for behavior and everything.
Yeah.
And clearly, this is an extremely powerful technique.
Yeah.
There is always a caveat, of course, because maybe you just introduce a new signaling pathway, which simply doesn't exist in an animal. And this signaling pathway, which you artificially introduce, will trigger behavior and everything is fine. But it's something which doesn't exist in life. But obviously, we have to have some technology to study it, and optogenetics is one of the very, very powerful technologies.
Yeah.
With all that, of course, comes another question, and I think it's a very important question. So calcium is a universal second message. Calcium goes up and down in more or less every cell around the planet Earth.
Yeah.
And as such, it has many generic functions. For example, goes to mitochondria, says, give me more ATP. Goes to the nucleus, says, I need more proteins, and so on and so on. And that is generic. It happens in every cell. We come to muscle cell, for example, and calcium has a very specific function. It initiates contraction. Yeah. Now, what is the specific function of calcium in astrocytes?
Don't know, right?
Well, of course, there are some ideas. So one of a very, very popular idea was a concept of the so-called GLIA transmission, in which people said that, astrocytes are as good as neurons, so they have vesicles which have glutamate, and they can release this glutamate, and blah, blah. And that happened all probably about late 1990s, and the first conceptual work was done by Phil Hayden and Alfonso Araque and Vlad Parpora, which is called tripartite synapse. And I think that was an absolutely seminal paper, which more or less breaks through glia into a neuroscience.
You know, Phil Hayden and I we were in the same lab when I was a postdoc with Stan Cater, Ben Cater.
Yeah.
A long time ago.
Phil is a very, very nice chap. I like him very much, and I like Alfonso, and I love Vlad. So it's, again, quite a camaraderie. But now when we start...
So I guess what's the role? So ATP, right, we think of as an energy molecule, that energy in these phosphate bonds and so on. But what about ATP as a signal between cells?
Oh, yeah.
That turned out to be really interesting.
That's another absolutely beautiful story, which connected to Jeff Bernstock, of whom I mentioned at the very beginning. And again, let me give you a little bit of history, which shows how coincidental is everything in this world. So ATP was discovered in 1929. And that was Fiske, Subarow, and Karlin. And at the very same year, 1929, St. Georgi in Hungary proposed the signaling role for purines. He proposed that adenosine is a transmitter or a hormone. Yeah? And Jeff Bernstock was born in 1929. And that was Jeff, who in mid-1960s realized that when he was looking at Taenia coli or some other peripheral junctions with sympathetic nerves, he can block everything. He blocks all adrenal receptors, all substance Y, whatever. And the response is still there. And he realized that it was ATP. And that created an outrage of the whole world, because everybody said, ATP is a molecule of energy. How can you, throw it away?
Right.
Took him 30 years, but that became a truth.
Yeah.
So ATP is a powerful, powerful neurotransmitter. And what is really interesting here is so if you take bacteria and you put ATP, bacteria will go away from it. How? We don't know. No idea. Because the very first receptor for ATP appeared in amoeba. So it's already eukaryotic. Now, practically every cell in the earth has at least one type of purine receptor, be it ATP or ADP or AMP or adenosine. So it's totally ubiquitous. And it's, of course, an absolutely natural danger signal, because every cell contains millimolars of ATP, 5 to 10, yeah? And if the cell is dead, all this ATP goes, and that's it. And so these receptors, I think, evolutionary, were probably survivalistic receptors, because you need to know. If you know that there's ATP around, it means that somebody is wrong, somebody is dead.
Right, or in danger. Yeah, so it's like a danger signal. My neighbor is under stress or
absolutely
dying.
And that is utilized by nature in some totally peculiar and amusing ways. So, for example, some lobsters, they can find out a prey, and lobsters are eating their scavengers. They're not eating fresh meat. They're eating dead meat. Yeah. They can discover it by hundreds of meters. Why? Because they have a special pair of receptors. One is sensing ATP, which is life, and one is sensing adenosine, which is death. And a KD for, I think, ATP receptor is 13 picomolar. Wow. So they can sense over many, many, many meters in sea water that there is something going on, and they know, is it dead or alive? And if it's dead, they go there and eat it.
Wow, very interesting.
Totally amazing. So life is, it's full of, I think, natural science is probably the most interesting part of our Earth, because it's so multifaceted.
Yeah, it's incredible how these things have developed over millions of years. Now let's shift and talk about aging and disease. You've done work on glial cells and Alzheimer's disease and things like post-traumatic stress disorder and depression. So maybe if you can spend some time talking about your work in that area.
I think it's another incredibly important issue, which probably, as I said at the beginning, when glial research started, it was all centered in pathology. Yeah. Because those cells are changing in pathology. So if you take nervous tissue, we have a lesion. Again, neurons can't do much. They don't have energy, they don't have blood. Okay, I'm dead. So what neurons usually do, they shut down in order to limit energy expenditure. What glial cells do, they actively change in order to protect. And the best possible example is what is called reactive astrogliosis, or reactive gliosis as such. It's a reactive change in glial cells in response to damage. And what happens in damage? When we damage our brain, it's more or less the same as we damage our skin. So we kill something, yeah? And this something is replaced by fibrotic cells, by fibroblasts, by pericytes, and they make a fibrotic scar. The difference between brain and skin is that in the brain, this fibrotic scar is surrounded by glial barrier. And this glial barrier limits the damage. It helps the damage to heal. It helps to make this scar, and it protects everything which is around. And then it starts actively contributing to regeneration. So if you interfere with those cells and kill the barrier, it doesn't regenerate, it doesn't heal. And that is, of course, its acute role. But then we have a whole class of diseases in which glial cells are changing. More or less, it's impossible to find a single neurological disease in which glial cells are not changing. Yeah. And more or less, in my view, because they are defensive cells, they try to protect. And when they are successful, we don't have a disease, nothing happens. If they fail, then we, of course, have a disease. And if you think about it, over our lifespan, which is what, 80 years or 100 years, whatever it is, In 99.9% of cases, these cells are successful. Nothing happens. We survive, we continue functioning. At the moment then it's an ultimate end of the life, which also has to come. So in that sense, I think that as therapeutic targets, those cells are probably of primary importance, because if we boost their homeostatic and defensive capabilities, we certainly can interfere with pathogenesis. Because as I said, in most of the cases, what happens, these glial cells, they lose their ability to support. And there is, of course, a whole corpus of papers, probably thousands of them, which are calling glial cells neurotoxic, yeah? They're not neurotoxic. They lose the ability to protect. The most neurotoxic cell in the brain is a neuron, because it releases glutamate.
Yeah.
If you think what I mean. Of course, in many cases, the defensive responses can go astray. It could be some feedback. It could be vicious circle. So very simple example. To cope with ammonium, yeah? Like hepatic encephalopathy, so the liver stops. And ammonium goes up. And of course, in the brain, it goes up. Ammonium is detoxified by astrocytes. There are two enzymatic cascades. One is glutamine synthetase, and another is urea cycle. But when urea cycle is working, it produces peroxide. And peroxide is pretty bad.
Yeah.
So in order to counteract it, the astrocyte should increase production of glutathione. But if astrocyte is stressed, then it doesn't produce glutathione, and we have vicious circle, coma, and death.
So there's many ways. So you've mentioned several ways now that the glial cells can protect the neurons, taking up glutamate, for example, or
yep.
Detoxifying, potentially oxidative damage, and they also produce growth factors that are good for neurons. My early work, I did work looking at effects of glutamate on dendrite outgrowth and survival of neurons during development. And as part of that, I was looking at different growth factors to see if they could protect neurons against the damaging effects of glutamate. And the one that was really protective is FGF2, or we called it basic fibroblast growth factor back then. And it's interesting because a lot of times you'll see in textbooks, the idea will be these growth factors are released from a cell, say an astrocyte, which actually produce a lot of this particular FGF2. And so the idea is they're released and kind of they diffuse all over. But we had this evidence actually when they're released, they tend to stay on the cell surfaces, and they mainly affect the neurons that are directly contacting them. So, yeah, so anyway, this is another way that so during aging or in disease, it's possible, I guess, there could be some compromise in that
absolutely
mechanism too. So.
Well, I think the aging is the most interesting thing, because of course, when we come to the aging and the brain, we have two pathways. One is what we call physiological age. And again, I know many people who live to their late 80s or early 90s who are completely intellectually preserved. And of course, we have pathological age. And clearly, some of this pathological aging is genes, so genetic form of Alzheimer's disease, what can we do? Some of them, I think many, are traumatic, so it's post-vascular, post-stroke dementia and everything. Some of them are what we call idiopathic, means that we don't know anything about it, and that is Alzheimer's disease and everything else. But still, it's not everybody. It's a part. So in a sense, that means that probably what we have this balance between defensive system and pathological burden. And as long as the balance is shifted towards defense, it's all physiological. When it goes other way, it's pathological. And what is most important, we can modify it. And I don't think it's a drug. I think it's lifestyle.
Yeah, I agree.
It's physical exercise, is food, is pleasure, is limiting stresses, is having positive emotions, you name it. So what is called quality of life.
And
What is
what
the
you and
most
I are
amazing?
doing now, Alex, engaging in intellectual
Absolutely,
and
absolutely.
social interactions. Then I'll
And
mention
what is,
one more.
can I just stop because this is really fun,
Uh,
really interesting. If you do these life modification styles, what happens is astrocytes which respond, they become larger and more complex.
huh.
So
Yeah.
that really is very interesting.
So there, I don't know if do you know Juan Troncoso?
Sorry?
Juan Troncoso at Hopkins. Anyway, he's a neuropathologist, and he had, some people, they're 90 years old and they're cognitively normal, really good. Their learning and memory is still really good, but then they die, and then they look at their brains, and there's a lot of the amyloid protein. And Juan found that in those people, and he looked mainly at neurons, but the nucleus of those neurons was bigger, and the cells seem to be bigger, so they're somehow responding adaptively to the stress of the amyloid. Probably similar with the glial cell, I guess.
Probably. Well, I mean, I very much wonder if beta amyloid is actually epiphenomenon in Alzheimer's disease. We don't know. I mean, the only thing which we know that is spectacular failure of every therapy against beta amyloid.
Yeah.
That's we
Yeah,
know.
the thing that, the geneticists, of course, point to the mutations in the amyloid precursor protein, but we pointed out that those mutations, so there's a normal function of this precursor protein. It's a
Oh, yeah.
membrane protein. The amyloid beta peptide is part of it. And there's this normal enzymatic cleavage that prevents, it cleaves right in the middle of the amyloid beta peptide, so it prevents its accumulation. That's called alpha secretase. And that occurs in response to synaptic activity. We think it's even this, we call secreted form of APP, has a normal neuroprotective function. At a Japanese postdoc, Kazutoshi Furukawa, who actually did some nice patch clamp work. He trained with Akihiko,
Ah,
who,
sorry.
Kazutoshi thinks Akihiko should have
Yes,
shared
I
the
know
Nobel.
Norio very, very well. He is in Kyushu,
Yeah.
which is the southernmost island. And for 10 years, I was teaching there every winter.
Wow.
It was beautiful. I
You
love
think
Japan.
he should have shared the Nobel Prize with him?
Oh, Norio? Well, no, don't get me wrong. I mean, I think that Sakman and Nair got it not only because of the pipe,
Yeah.
but because of lots of things associated.
Yeah. Yeah.
Because of secretion from the point of view of Irving and all the molecular biology of receptors
Yeah.
from
Yeah.
there. So it was not just
But anyway,
uh,
get back to
no.
this. So this Kazutoshi found this. He did, patch clamp, perforated patch clamp.
Mm-hmm.
But anyway, the bottom line, he found this secreted form of APP activates a potassium channel, probably the so-called maxi K channel, and so it hyperpolarizes the neurons. So it's kind of maybe a very rapid kind of negative.
Mm-hmm.
So glutamate's released,
Yeah.
and at the
And
same time, the secreted APP is released at the synapse.
so it's an immediate negative feedback.
Yeah.
That's very cool.
Yeah. Well, Nature paper.
I think there are also some evidence that beta amyloid is a part of anti-infection system of the brain.
Could
I
be.
mean, I don't know how much, but there are. So in a sense, what we know is that if we delete APP, the whole thing doesn't work.
Yeah. Yeah. And let's get you. So you mentioned the lifestyle and the importance in keeping the brain functioning well and resistant to disease during aging. And you mentioned exercise. We talked about intellectual challenges.
Mhm.
And then you actually, I think you were on a paper with someone did caloric restriction and then looked at.
Yes, so we did it together with my colleague Alexei Semyanov. So we did caloric restrictions. It's a part of a big story, yeah? So this caloric restriction was made in young animals.
Yeah.
And we are going to do the same in very old animals, but this is still under progress. And in parallel, we did also high-fat diet in young animals and in old animals. So in young animals, high-fat diet is actually positive. In old animals, yes, in very, very young animals, that increases.
Oh, very young, yeah.
In old animals, it's very negative.
Yeah,
we know that. So it causes all sorts of mischief. With caloric restriction, well, give me another year, please.
Okay.
It's quite complicated, yeah.
Yeah, I'll be very interested to see what happens with that. Now, okay, so far you've talked a lot about astrocytes, you know, how they're kind of, they have these sodium and calcium changes that seem to affect the function of the astrocytes, and in turn, through multiple mechanisms, affect neurons in good way, facilitate their functions, support their survival and resistance to stresses of aging and disease. And now, astrocytes, we haven't talked about this yet. It's very interesting. So, it turns out, apparently, human brains have relatively more astrocytes than rodent, or not? Or well, definitely, yes. There's good data that they're more elaborate. Their structures
they're much more elaborate. So okay. that's another very, very interesting thing, which dates back to Friedrich Nissl at the end of 19th century, who sort of made this notion that we are so intelligent because we have lots of glia, although he wasn't even sure whether it's glia or not. Anyway, it became extremely popular. And if you go for one of the Eric Kandel classical neuroscience books, it's written actually that there are 10 to 50 times more astrocytes. It depends. Yes, it depends on which edition, of course. So I read it in about 2002. There are about 10 to 50 times more glial cells than neurons. Yeah, this is, of course, totally wrong. No. The total numbers is more or less 1-to-1.
Yeah.
There is quite a big original difference. So, for example, in cerebellum, which is full of neurons, most of our neurons are in cerebellum, there are very few glia. In brainstem, indeed, there is much more glia than neurons. In cortex, and that is also quite interesting, the largest subpopulation is actually oligodendrocytes. It's not astrocytes.
Huh. And
that probably makes sense because if you look into the difference between human brain and rat brain, in human brain, 53% is white matter.
Yeah, okay. Yeah.
In rat is 12%. And in gray matter, there is plenty of myelination goes on, and this myelination is plastic.
Yeah.
Something is myelinated, something is demyelinated, depending on everything. So
that makes sense. So these oligodendrocytes are the insulation on the axons. The human brain, it's a bigger brain, so neurons have to have longer axons to reach across. Yeah.
Precisely. But if you go with numbers, the highest number of glial cells is in whale or elephant. Because the brain is huge. The brain of a whale, minke whale, is 7.5 kilograms.
Yeah.
It's of that size. And it's only 12 billions of neurons. 90 billions are astrocytes because you need to fill the space somehow.
Interesting.
But what is really interesting, if you take a single cell, the whale astrocyte would be like rodent astrocyte. And human astrocyte will be of that size and complexity.
Yeah.
So what happened in evolution, at least in some area, I'm not talking it happened across the brain. So let's say striatal astrocytes are very different. But in cortical astrocytes become much more complex. More than that, human brain has several types of astrocytes which do not exist in anything else but highest primates, and that are called interlaminar astrocytes. We have no idea what they are. The soma are sitting just beneath the pia, and then they send very long processes, like a couple of millimeters, which penetrate all layers of the brain, which go to layer 5, sometimes even to layer 6, usually layer 4. What they're doing, we don't know. But if we take Alzheimer brain in BRAC6, these astrocytes are gone completely.
Are they, okay, so during brain development, you have these radial glia that they're like on the ventricle and they extend up. Mm-hmm. And these cells you're talking about they're kind of structured, but their cell body is different.
Yes, they have this sort of radial morphology. I mean, radial glia is more or less neuroepithelial cell, yeah?
Yes.
And what distinguishes radial glia, proper radial glia? They have two processes. One terminates at the ventricle and one at the peak. And when the brain goes thicker and thicker and thicker, so when we are born, it's about 2 millimeters. So these radial glia are literally 2 millimeters, but that's probably the end of it. They can't do it anymore. So they disappear. And they have a progeny. So in some parts of the brain, we have cells with radial morphology, like Bergman glial cell or Muller cell or Tanicytes, so the name. What is the origin of these interlaminar astrocytes? We don't know, because how you are going to do fate study in human brain? Yeah. And in rodents, they don't exist. As simple as that.
Yeah. But well, maybe these, there's a lot of labs now are making these so-called organoids or brain assembloids where they get that, get kind of, and they're doing it from human, right? Because they take fibroblasts, the iPSCs, induced pluripotent stem cells, convert them to different cell types and so on. So there's maybe some of that work will eventually help solve that.
Of course, it's a huge progress, because what we need is to humanize the research, if you will. We have to realize that translation from a mouse to the human, especially in pathophysiology, is non-existing.
Yeah, that's right.
So we spend, I don't know how much time and everything on, let's say, animal models of Alzheimer's disease, yeah?
Yeah.
And don't get me wrong, I spent 20 years of my life with it. And I think it's important because we did something, we learned something, but the most important thing we learned, it is not translatable. So we need to do something new to go to human brain and to study it. And of course, stem cells, organoids, everything, this is a big step forward. But eventually we will come to a ceiling again, because this is in vitro system.
Yeah.
And we have chronic disease which lasts for 30 years. Yeah. How we are going to model it?
Yeah.
I think it's sad, but it's reality.
Yeah. And all these diseases, particularly Alzheimer's, Parkinson's, Huntington's, ALS, by the time person's symptomatic, the disease is so far progressed. And then one problem with animal studies, many animal studies, they'll give the treatment like early on before there's much, so they're really looking at, prevention or protection rather than trying to recover from.
Absolutely.
Yeah. Okay, so why don't you mention, we can finish up, I guess, talking about microglia and
oligodendroglia.
Yeah.
Right. Oligodendroglia we already touched, because it's white matter.
Yeah.
And indeed, as we discussed, white matter is what defines human brain. Now, historically, evolutionary, what was that? It was the insulation, which not only insulated the axon, but it also sort of hoarded major excitatory channels like sodium channels into the nodes of Ranvier.
Yeah.
So when we have an axon, we have this internodal region, which is not conductive, and nodes of Ranvier. So that is what is called saltatory conduction of action potential. Of course, nobody jumps, but channels are open because of electromagnetic field. And what is the most amazing, it is generally believed that this is only in vertebrates. That is not the case. So many non-vertebrates do have myelin without myelin protein, but they have a very similar structure. And the fastest transmitting axon in the Earth is a prone. It's a Japanese prone or Dublin prone, you know, this langoustine, which is very tasty. It is up to 200 meters per second because it has most elaborated thing. It has like axon and periaxonal space and then many, many layers of these glial cells, yeah? There is no myelin protein, but it doesn't matter. So it's like axon within an axon. And that is okay, this is something which always strikes me when I look into biology and evolution. The very first predator on Earth was a giant prawn back then in Cambrian Sea.
A prawn, a shrimp-like.
Yes, a shrimp. It was 1 meter long, and it was the only predator.
And they can move really quickly.
Yeah, but that is even more interesting. It had, like arthropods have, external eyes, yeah? So it's a composite external eye, which has about 100,000 elements. And each element has an axon which feeds it to the brain. So you have 100,000 axons, and you need to feed it at the brain, and they have to be fast. And what is an answer of invertebrate for the speed is to increase diameter. So the loligo, the squid, has 1 millimeter.
Yeah.
Now imagine 100,000 axons, 1 millimeter each. Impossible.
Yeah.
So the solution was make this myelin and they become very thin. And that, I think, what was a major evolutionary drive is to make it small. Because if you think about energy, for example, of course you save on energy because you have only nodes of Ranvier, yeah? But imagine how much energy you need to produce myelin all life long. It's even more. So in my view, it's all to make the brain small and effective. This is oligodendrocytes. And these oligodendrocytes are quite interesting developmentally because they originate from their dedicated precursors, which are called oligodendroglial precursor cells. And those cells are very interesting because they populate the whole brain rather homogeneously. It's about 10% of all glia, and they are proliferative. So they proliferate all the time. They self-renew, and some of them probably die and disappear, and they're always there. Some of them produce new oligodendrocytes to remyelinate things. That's no doubt about it. But many of them don't. What they do, we have no idea. It's totally enigmatic cell, which I really, truly love.
Yeah.
And of course, when we are coming to microglia, it's another very interesting thing because this is the most evolutionary conserved neural cell ever. So the first microglial cell appeared in leech. And if you look into morphology of leech microglial cell and human microglial cell, you probably wouldn't tell which is what. They're identical. And if you go through all evolutionary ladder, it would be identical throughout of the species.
And throughout all this time, so these are they're part of the so-called innate immune system.
Yes, it's innate immune system, yes. So it's fetal macrophages which enter neural tube very early, like I don't know, for human it will be probably end of 6 weeks of gestation. They enter and then they change. So macrophage is amoeba, yeah? Microglial cell is neuron. So it has a very small cell body and has many, many processes which are very elaborated, and the processes are moving.
Yeah.
They're physically moving through the brain. So it's the most amazing cell. So I wouldn't even call it macrophage anymore. It's really a neural cell of non-neuronal origin.
Yeah, I remember I got my PhD at University of Iowa, and I went back there and gave a talk. I can't remember when, and I don't even remember whose lab it was, Smith. Anyway, it showed me this beautiful video of time lapse of microglia moving. They're just they're like surveilling the brain, moving around, looking for potential whatever bacteria or maybe degenerating synapses that gobble them up. Yeah, they're really fascinating cells. And as you mentioned early on, that they get a bad rap, right? Because they can produce these so-called pro-inflammatory cytokines, but on the other hand, I mentioned we did these experiments where we showed that certain growth factors protect neurons against excitotoxicity. And then I had a postdoc, he started looking at inflammatory cytokines, tumor necrosis factor, interleukin-1 beta, and we expose the neurons in culture to those and then wait a while and then add glutamate at a concentration that will kill a lot of the neurons. And surprisingly, we found that tumor necrosis factor, TNF, protected the neurons against excitotoxicity. And moreover, we found this was by activating a transcription factor, that is a protein that regulates gene expression, called NF-kappa B. And so then we had another PNAS paper following up. And we had very hard time getting these papers published because Rivera said, well, no, TNF is bad, or NF-kappa B has been associated with bad things. But it turns out that this is an adaptive stress response pathway that the cells are naturally using to protect.
Oh, Mark, I can't agree more. And again, if you look into evolution, evolution never, ever chooses anything which is detrimental.
Yeah, that's right.
It always chooses something which is either neutral, which happens all the time, or positive and beneficial.
Sure. Yeah.
And every system which exists in our body is actually aimed at preserving homeostasis through allostasis, through the change, and to protect. And of course, in certain conditions, it can acquire deleterious proportion, no doubt about this. But in most of the cases, it's inflammation as such is the most protective process in the earth. Without inflammation, imagine you make a scratch, no inflammation. It will, of course, be full of bacteria and everything, and you will die in 3 days. Yeah. The inflammation is a central part of our survival. Yeah. Like pain is central part of our survival, and so on and so forth. And I think, unfortunately, what is happening, there are lots and lots of groups which, and now, of course, you know what science is today. You have to produce and produce and produce and produce, and you have to be within the mainstream. And in very many cases, people don't really spend some time to think and to reflect and to understand what's.
Yeah, that's a good point. Yeah, so, and one thing I did throughout my career, I'm interested in, broad, like the big picture of things and how things fit together. So I'd go to cell biology meetings and I'd see what's going on in these research on these other cell types. And what's the implications of that for neurons? And you're right, a lot of people now, for example, Society for Neuroscience is a good example. They have an annual meeting each year, huge, 30,000 people. And a lot, I think you're right, a lot of scientists at all levels, even, they'll go to their own specific, very specific area and they'll interact with maybe a dozen people in that specific area, and they don't pay any attention to anything else at the meeting, and think about, hey, well, to learn for the, just learning is important, but then also, the more information you have, the more ideas you can come up with.
Absolutely.
New ideas. Yeah.
Well, I think that probably reflects very much the whole change in academic world, because again, when I was a young student, who was caring about impact factor or other crap?
Yeah.
It was non-existing, You spend a year, you make a story, you present the story, your peers say, that's a reasonable story. You publish it in Journal of Physiology and everybody is happy. Now you have to produce hundreds of papers and they have to have this impact factor, which is, in my view, it's a crime.
Yeah.
I mean, plus now we have all this open access rubbish with all these thousands of predatory publishers, which are, they produce not even noise. Noise is neutral. They produce detrimental noise.
Yeah, what Alex, what you're talking about here, for people who aren't aware of this, that aren't scientists. So you and I, probably every day, we receive emails from this, what you're calling, and I agree, predatory journals, where they're essentially, they publish anything, and they're not critically reviewed. And you and I, for the most part, know this, and just, we can't really pay any attention to those papers because we know that they're not likely to be reliable. Yeah. But nevertheless, a lot of things are getting published, and yeah, it's crazy. Anybody can publish anything now, it seems like.
Precisely.
But the leaders in the field know, understand, what's, for the most part, the.
Oh, absolutely. But Mark, I mean, this is very dangerous for new generations.
Yeah.
Because they're overwhelmed with this incessant stream of something which is not even information, and they don't know how to distinguish. So that is where mentorship is very important.
Yeah.
There is when example is very important, and then where you need some suggestion from your elder peers, if you will. That is really important.
Yeah.
So I think that one of the duty of scientists, and certainly I consider it my duty, is to spread education and to help young people as much as you possibly can.
Yeah. Yeah, and we need this, a lot of, if you're going to be a scientist, you cannot be too keen on making a lot of money. You have to be,
nope.
But it's a fantastic thing to do, because your whole life you're learning new things, you're discovering new things, and
you are free.
Yeah.
Because can you imagine sitting in the bank for 8 hours a day and then having 2 weekends? That would be the end of my life. I would commit suicide.
Yeah, or lawyers, oh my gosh.
Yeah, agree. Well, dear lawyers, don't get offended, but.
Well, no, but it's when you get to these things where people are trying to make a case with essentially data that's either misinterpreted or unreliable data, and then trying to convince people of something when the person themselves may not even be confident that what they're trying to convincing them is true. We can avoid all that things as scientists. Yes. I think that's good.
Yeah, I mean, but at the same time, as you said, science is probably the closest you can get to the act of creation, because understanding is more or less creating the universe inside yourself.
Yeah. Yeah.
And that is exciting beyond belief. So I can't imagine any other, probably being a musician like Bach or Mozart would be the same, or artist or something, or poet, you name it. But this is act of creation, which I think, I'm very happy that I have this career, and I never ever thought about anything else.
Yeah, me too. It's wonderful. Okay, Alex.
On this
Well, I
optimistic
know,
note,
this topic was maybe a little too broad, but it's a good introduction to GLIA and so on, and maybe in the future I'll have some more
with big pleasure, Mark.
focus thing.
Anytime. I enjoyed this, whatever, 1.5 hours. Wow, I didn't even realize.
Yeah, see, that's when you're having fun, it's goes quickly.
Yes, absolutely.
Okay,
Thank you very much.
by the way, and you probably haven't come across this person in Manchester because she's in a very different area, but there's a clinician named, actually, Michelle Harvey, who we collaborated with her on some human studies of intermittent fasting.
Mm-hmm.
Not looking at the brain, but looking at other things. And she's like focused on cancer.
Oh, she's probably in Christie Hospital, yeah.
Yeah, I think so. But finally now we have been doing some studies, and we're writing up a paper now of looking at some effects on fasting in people at risk for cognitive impairment because their age,
Yep.
55 to 70, with obesity and insulin resistance. And we have a baseline, then 2 months after being on intermittent fasting or control. We do battery of cognitive tests, structural and functional MRI. We got CSF samples. We got blood. We even did some MR spectroscopy to look at certain metabolites and some interesting findings. So stay tuned for that.
That's really very interesting. Just curious, did you do PET? Positron emission?
No, we didn't do PET.
There is a wonderful thing
PET
which
with
is called
radiolabeled glucose or what?
No, with deprenyl. Deprenyl is a molecule of happiness, of course. It's a blocker of monoamine oxidase B.
Yes.
And in the brain, 90% of this MAO-B sits in astrocytes.
So it's involved in
an interesting switch.
It's involved in metabolism of the monoamines, the dopamine, yes, the serotonin, yeah, norepinephrine. Yeah. So another important function of astrocytes is picking up those. So anyway, if you so deprenyl inhibits those, it'll increase serotonin.
Yes, it increases bioavailability, yeah, of catecholamines in the brain. Yeah. And if you look into the aging, and especially in Alzheimer's disease, locus coeruleus is extremely vulnerable.
Yes.
So we have a decrease in catecholamine presence in the aged or neurodegenerative brain.
Yeah.
So that is quite interesting.
That is interesting. Different. Huh. I'll have to talk to the neurologist, my colleague, yeah, who's doing this about that.
Yeah, it's a classical PET marker for astrocytes. It is labeled with carbon 11.
Yeah. Okay. Yeah, they have Hopkins, of course, they have a.
Oh yeah, they of course have it. Yeah.
Yeah.
No doubt about
it. Okay, Alex. Well,
Mark, thank you very much. I enjoyed this time very, very much.
Yeah, I did too. I'm glad I invited you. It's good to see you.
And good to see you too. And good luck. And when it's going to be aired?
We're all wishing Ukraine the best, and your yes, colleagues back there.
For the, I mean, yes, we shall win. Thank you.
Bye, Alex.